Okay, for the next talk, do you hear me? Yes, okay. I have the pleasure of introducing Maryam Vaziri. Maryam is an assistant professor at the University of Delaware, leads research in the Department of Psychological and Brain Sciences, focused on the neuronal mechanisms underlying real-time interactions with objects and people. Maryam holds an MD from Tehran Medical University and a PhD in cognitive psychology from Harvard University. Her current projects delve into object recognition, affordances, and action prediction, combining methods such as neuroimaging, body movement tracking, and machine learning. Her work links cognitive, social, and motor neuroscience to a deeper understanding of how we interact with the world. Floor is yours. Thank you so much.
Okay, hello everybody. Thanks for inviting me and thanks for all the great talks. Okay, shout out to Madine. These are YouTube videos of people wearing head-mounted cameras going about their lives. This is the reality of vision. This is how vision is in daily life. It's not a series of snapshots that we are going around labeling objects in it. It is actually moving about the environment, seeing stuff, trying to figure out how to position your body to interact with things, interact with objects, and interact with people. In this spirit, my research program sort of tries to get vision out of the screen and study vision in the context of real-time interactions. And I'm going to give you some highlights of my research in two different domains, interacting with objects and interacting with people. In the domain of interacting with objects, I'm going to focus on a particular behavior of grasping objects. Why grasping? Because it is one of the most common ways we interact with the environment, because we have a specialized hand that is very unique in the animal kingdom. Okay, let's kind of really zoom out and think about the computational problem. You see this teapot, some processing happens in the brain, and you get an output. This output could be the category label of the object. So you can think of this as a multidimensional embedding space, and the teapot occupies part of that space. Now you want to grasp the object. Now you have another embedding space, which is the shape of the hand at the end of the grasp movement, right? These two are two different behaviors. Now think about these two other objects. Now you have a cup with a handle and a cup without a handle. In the category output space, the two cups are next to each other away from the teapot. In the grasping output space, the two objects with a handle are next to each other away from the other cup. So you have two output spaces that have some overlap. They both rely on processing of the shape. They rely on processing of the visual information, but they have some overlap and some distinctions. So question is, can the same visual process actually support both of these tasks? How do we answer this question? To answer some question like this, we basically have to start by behavior. We really need to understand these two output spaces. We need to map out multiple behaviors from participants. And then we can actually, after we learn about these outputs, about these behaviors, about these spaces, we can look into neural mechanisms and figure out what parts of the brain are involved in it. So to do this, I developed a data set. I called it Things in 3D. It's part of the Things Initiative. And basically, these are 3D printed objects that have meaning for participants. People know what they are, but they allow us to study multiple behaviors. They allow us to ask about them, about the similarity, about the object category, but they also allow us to look at grasping movements. So we collect two different behaviors from these objects. One is the behavior of grasp movements, and the other one is we ask people to do similarity judgments on these objects. To do grasp movements, we basically connect sensors to people's hands and ask them to perform, grasp movements. I think I have to, how do I play the video? Okay, there we go. So this is an example grasp movement. Let's move it. There's some Keynote to PowerPoint translation, that didn't quite happen. It's okay. So these are sample grasp movements. This is the actual data, it doesn't really matter. So we have sensors attached to the fingers of subjects, and we have grasp movements. So the shape of the hand at the end of the grasp is basically forming to the shape of the object. It's a proxy for that part of the objects that is relevant for the grasp. We get these grasp movements, we identify the end of the movements, we have 16 sensors, XYZ position, that's 48 numbers. We correlate those, and we get a similarity matrix. Some objects are grasped similar to each other, and some objects are grasped differently from each other, okay? We can look at this multidimensional space, do dimensionality reduction. 6 dimensions explain more than 90% of the variance in this space. And when you look at the layout of the object in this space, it is related to the shape of the object that is relevant for the grasp, the orientation and the size of the object that plays a big role in this space, okay? Now we ask subjects to do similarity judgments. We take triplets of objects, put them in front of subjects, and we ask them to pick the odd one out. Okay, and we do this over and over again with lots and lots and lots of trials, and you get another similarity matrix. Okay, you have now 2 similarity matrices. This one is based on how similar they are overall, without any instructions, okay? For this space, it's much more higher dimensional. It's 15 dimensions explain more than 90% of the variance. And when you look at it, the semantic information, the category of the object plays a big role in how they are placed in this space. Tools are next to each other, musical instruments are next to each other, toys are next to each other, okay? So you have 2 spaces. How similar are these? They both rely on shape. They both need vision, okay? How similar or different they are. The correlation between them is 0.24. Both of them are highly reliable, 0.8 reliability. The correlation is 0.24. So you have 2 outputs that are really distinct from each other, suggesting that if probably for producing these 2 outputs, you need to diverge the network at some point in your process, right? Okay, so we basically have 2 output spaces, 2 distinct outputs, and we want to know what part of the brain are involved in creating those outputs, right? Visual system. Where would we look? The way we think about visual processing is very much shaped by this view put forward by Mishkin and Unger later, that visual information is processed in 2 separate pathways. One is the what, ventral pathway, and the other one is the where, dorsal pathway, okay? So this is a meta-analysis of all the papers that use the word object, form, shape, etc. All of the regions that show up with high probability in these papers is very much telling us the story of ventral dorsal stream, right? There's a lot happening in the occipitotemporal and ventral stream, but not much happening in the dorsal stream. That's the parietal cortex, the dorsal stream, right? Is this real? Or is this because the literature is actually biased by the dorsal ventral pathway hypothesis? To answer this question, let me show you one little video. I mean, there's some work that has been done that shows that there is some object shape responses in the dorsal stream, but instead of reviewing the literature, I'm going to show you one patient video. This is a patient that is object agnostic. They cannot recognize objects, okay? They're shown a picture of that combination link in the beginning of the video and asked to identify the video. In the video what the object is. And they say, we don't know what it is, they have no idea what the object is, okay? Then the interviewer asks one more question.
Or something.
Show me what you do with it.
Okay, that part's gonna go up here, and now that ain't the way that works. But that's what I'm doing anyway.
Yeah, what are you doing with your hands?
Well, I'm not sure. Just a minute, let me look into me. Okay. Damn. That's weird. Let's see. Now that part's gonna come up here, and this'll come out.
Okay, so the patient has no idea what the object is, but they keep doing the action they would normally do with the object. They have some idea of the shape of the objects. Their ventral stream is damaged. They can't recognize the objects, but they know the shape of the objects. They know what they can do with their hands with the objects. And actually, later down in the video, they identify the objects by looking at their hands. They sort of deduct what the object is based on their hand movements, based on their body movements. So what does this tell us? That you can have damage to all those regions and still have some information about the shape of the object, right? So what's going on? Is there nothing in this sort of empty area? So I set out to actually look at this, starting with simple experiments showing videos on images on the screen, right? So we show 8 categories of objects, right? And 10 images per category. And we basically use a block design paradigm. We show these images in a block design paradigm. We localize a whole bunch of regions in the dorsal stream, in the ventral stream, in all retinotopic maps in the early visual cortex and all throughout parietal cortex. And what we do is basically we get the responses to each voxel in the brain. We get data from each region in the brain. We do a pairwise classification. Luckily, I don't have to explain that here in an AI conference. Everybody knows what that is. So we train the classifier with some data, test with some left out data. We have multiple pairs. We average them all together. We get average classification accuracy for all these objects. And we want to look at the classification accuracy in multiple regions in the brain, right? Okay, so we do a couple of more things. We remove the differences in average luminance, contrast, and spatial frequency. Low-level features are not causing the problem, causing the classification. And we show the images in 2 separate positions, either in the upper visual field or lower visual field. We train the classifier in one position and test it in another position. We want to see if the classifier, looking at the pattern of activity in this place, can generalize across position. This is really important because we're looking at dorsal stream, which we know cares about position. And we want to see if there is a stable object selectivity in this region across changes in position, okay? So here I'm plotting the average classification accuracy. Chance level is 0.5. Early visual regions don't have much above chance classification accuracy. Ventral regions have good above chance classification accuracy. We also have above chance classification accuracy in many of our dorsal regions. Not as good as the ventral regions, but we do have them in the dorsal regions. I have replicated this experiment. So we have position-tolerant selectivity for objects in the dorsal regions, in parietal regions that we should not be seeing, basically, based on the classical hypothesis. I've done this experiment across changes in size, changes in spatial frequency, changes even in attentional task, and many others have found similar results in the dorsal stream. So we have shape responses in the human dorsal stream, which we still don't know why they are there and why they're happening, okay? So I've been showing you static images, but when you move around the environment, you don't just see static images. A lot of the objects we see around us are moving around, and the movement of the object conveys information about what they are. So when we move around the environment, we see static information and dynamic information tangled with each other from the objects. So are the dorsal and ventral stream object responses sensitive to dynamic cues or static cues of the objects? So to answer this question, we need to isolate the dynamic cues. How do we do that? We get videos of actual objects, 6 categories of objects. In these videos, we extract optic flow information, project that into random dot moving patterns. So we have only taken the dynamic information from the videos and projected it on random dot kinematograms. Here's an example video. Does everybody identify the animal in this video? Yeah? If I stop this video in the middle, every single frame of this video is just random dots. The reason you're seeing this object here is because there is movement in this random dot patterns that follows the movement of the giraffe, and you're able to see the giraffe. Again, we select a whole bunch of regions in the lateral occipitotemporal, ventral occipitotemporal, inferior IPS, and supramarginal geniculus. These 2 are in the dorsal stream. Those 2 are in the ventral stream. Here's the classification accuracy in dorsal and ventral regions. We have a whole bunch of other regions in this paper, but I've removed them for the purpose of the talk. Please feel free to read the paper. But you can see that we have above chance classification accuracy in both dorsal and ventral regions for both the static and dynamic stimuli. There's a little bit higher accuracy in LOT and VOT for static images, and in SMG, it seems to be a very high accuracy for dynamic stimuli, but it's not like there's dorsal stream only cares about dynamic and ventral stream only cares about static. the information is tangled across the whole brain in both dorsal and ventral stream. So we have object selectivity in the dorsal stream that, that basically responds to both static and dynamic information. So what is happening? Is this redundancy? Is there any difference between the responses of dorsal and ventral stream? Let's go back to this classification matrix. There's more in this classification matrix that I have been showing you. I've been averaging this matrix, but you can actually leverage this classification matrix and compare the responses in the two different regions of the brain. You can correlate these matrices and get a sense of similarity between regions. You can do that across a whole bunch of regions, and that gives you a 15 by 15 similarity matrix. This is now a 28-dimensional matrix under the hood that in this matrix, regions are basically occupying different parts of the space. So let's do like multiple dimensionality reduction on this and plot the first two dimensions so that we can visualize it, okay? This is when you plot the regions in early visual cortex all the way to the ventral stream, V4, LOT, and VOT. You kind of see the occipitotemporal hierarchy emerging from this analysis. I did not put position in this analysis. This is based on the way these regions are responding to these different kinds of stimuli. This is about what these regions care about in the stimuli, okay? And this is what you see in the dorsal regions. So you kind of see the divergence between parietal and occipitotemporal regions in this analysis. Parietal responses are not similar to early visual responses. They are not also similar to the ventral responses. They somehow diverge from these two sets of regions. Okay, so what so far I have shown you that we have like object selectivity, very good object selectivity in the dorsal stream, and that the responses in the dorsal stream are different from those in the ventral stream. And the question I'm after, which I have teaser trailer for it, not an actual result, is I'm trying to answer what is this difference and is this related to object interaction. So we go back to our large database of 3D printed objects. I've built this setup. This is what is happening in my new lab, still ongoing, to put these objects inside the MRI scanner and have people actually look at these objects and grasp the objects inside the MRI scanner. So they look at the objects, and a few seconds later, they hear a signal that says, go and grasp the objects. I'm interested in the visual section of the of the trial, but the plan is to see when people are in the context of grasping whether we can tease apart the responses in the dorsal and ventral stream and see if they are related to those two behaviors. We have now two different outputs, the grasp movements and the odd one out similarity, and we want to see if the responses in each of these regions relate and how they relate to each of these behaviors. Okay, so just to have like an in silico sort of version of this experiment, basically I use deep convolutional neural networks. So we have two different behaviors. We get a whole bunch of available, already trained deep convolutional neural networks trained for object categorization. Then we try to predict each of these behaviors from different layers of deep convolutional neural networks with just a simple regression. How can we predict the two behaviors using a simple linear mapping going from each layers of the CNN to each of these outputs? Okay? This is predicting the odd one out. So the y-axis is how well you're predicting, and each one of these is different networks. This is how well, so these networks are only trained for categorization. So none of these two behaviors are what the network is trained on. So we can predict the odd one out behavior from layers in AlexNet, and it increases as you go higher up in the hierarchy of AlexNet, and that's kind of the same across the board in all of these networks. Interestingly, when you try to predict the grasping behavior, it gets better up to the mid layers, and then it has a dramatic drop. So, which basically means that the later layers of these networks, which it's not too surprising, which are trained for categorization, are getting away from what would be useful for getting into these behaviors, basically. So it becomes less and less useful for predicting grasping behavior, which kind of suggests that really the network kind of has to diverge at some point in the hierarchy if we want to predict, if we want to have a model that produces both of the outputs. But I mean, really, this is just a testing bed. What we really need to do is actually run the fMRI experiment. So in the domain of interacting with objects, we have evidence that maybe we have actually two systems in the brain, one dealing with interacting with objects and another dealing with semantic understanding of the objects. And we have some preliminary evidence from deep networks to also add to that. But I want to sort of switch gear in the next, like, I don't know, 5 minutes that I have, and really quickly tell you about a little bit of work that I am doing dealing with interacting with people. And I'm going to again focus on a simple behavior of reach prediction when people are interacting with each other. And again, I'm going to start from mapping out the behavior. So we want a paradigm in which we can actually look at people predicting each other's reach movements. So I built this setup. Two people are sitting in front of each other. There are two targets on the screen, and they have to play this game. So one of them is the attacker, has to go to one of the two targets. The other one is the blocker, has to go to the same target as fast as they can. I kind of like demos, so I want to have you guys play with me. Okay, so put your finger in front of you. Imagine there are two targets in front of you. I'm going to go to one of the two targets, and you have to go to the same direction. Ready? Ah, good. Well, the subjects were told not to trick each other. But this task, as you can see, is pretty easy. You're really quick at doing the task. And it's surprising how well you can actually figure out where I'm going in this task, which is kind of what I found, actually. So we are recording the finger position of the subjects using sensors. We have these dots that are moving on the screen too, and this is our control condition. We have the dot that is moving, and subjects are actually playing either against a human or against a dot. This is the reaction time playing against a human. This is the reaction time playing against a dot. They are about 100 milliseconds. This is a lot of difference in reaction time, and it's a very robust effect. What is happening here? Maybe there is information in the body that is present that tells the participant that is in front of them, that sort of tells them which direction this person is going. Is that what is happening? To answer this, we actually video recorded the attackers when they were playing the game. And we have now videos. In the videos, we can go and remove every frame of the movie before the finger lifts off the table, basically. So we have now two conditions. One of them, you can see the full video, and in the other one, we replace all of the frame of the movie. We remove all of the predictive information before the finger lifts off from the table and replace it with a static frame. Okay? So all of the information is removed. These are the two videos. They're very hard to tease apart. They're different, but they're very hard to tease apart. So let's look at how reaction time changes. This is playing against a real human. This is playing against a full video. This is playing against a cut video. The two videos that you could barely tell them apart, they create 100 milliseconds reaction time difference, which tells you that there is preparatory movement, and this is kind of, sorry. That's okay. So there's preparatory movements in the body of the attacker that gives away which direction the attacker is going. And I actually tried to do an experiment to figure out where this information is located. Is it in the eye? Is it in the wrist? Is it in the trunk? Where are they located? So we actually had them play against each other while the screen was covered with different pieces of cardboard. So very, very low-tech experiment. And we also gave them sunglasses in one condition. This is when they see the whole body compared to the dot. They are fast when they see the bottom part of the body. They're fast when they see top part of the body. They're fast when you give them sunglasses. You really have to cover a lot of the body to get them to slow down. So that tells you that this information is really distributed throughout the body, and people are really good at picking up this piece of, like small pieces of information. This is actually, again, machine learning thing that we did to try to figure out a heat map of where the information is located. And basically, it shows you that the information is distributed throughout the body. So this kind of shows you that you have this knowledge of the kinematic movements of the participants. You kind of know how the body moves and use that to predict people's actions during real-time interaction. So where is this kinematic information located in the body? So to answer that question, I'm just going to really briefly mention one study that we used, videos like this. These are like point light displays of human actions that for some reason, we had like a PowerPoint keynote transition problem that destroyed a lot of my videos, but that's okay, we're gonna move on. We showed like point light videos of human actions. There are like 10 different kinds of actions. Again, we show stimuli either in the upper visual field or the lower visual field. We want to see if we find representations of actions that generalize across position. And this is a heat map of all the regions that show up in this analysis. We have regions in the dorsal stream, in the parietal cortex, and in the occipitotemporal cortex that have above chance classification for these 10 different classes of objects. And again, just a teaser trailer, please read the paper if you're interested, that we found that the responses in the occipitotemporal cortex related to the semantic similarity between different actions. What action is similar to the other based on people's subjective judgments. And regions in the dorsal stream had responses that were more related to the actual objective movements in the video. What body part is moving? Is there hand moving? Is there feet moving? Please feel free to read the paper if you're interested in this. So again, we find two separate subnetworks in the human brain that processes action for probably different purposes. So again, we have evidence of these two distinct networks, both for static objects, dynamic objects, and for human actions in the human brain that suggests that maybe we don't have just one single hierarchy in the brain that processes visual information, and if we want to get into different kinds of outputs, different kinds of behaviors, we might actually need to separate networks into multiple pathways. And just to go back to our dynamic stimuli, what I think basically the spirit of the talk is, and what I want to convey is that we really have to get out of in front of the screen, static images, and try to think of, we learn a lot from those, but we have to try to think of paradigms that will let us systematically study vision in the context of real-time interactions with the environment. And that is, I think, doing that, we can actually build understanding of the brain and models of the brain that relates to everyday interactions with the environment more. And this is all the people that helped me with this. I can't name all of them. And thank you for listening.
Really running out of time. So, okay, I do have a question. There's no one else. Okay, so, main neuroscience AI. What do you think is the message here for AI models, for vision AI models? There's a lot of focus on object recognition, ventral type static models. What do you think you should take away from all these two-path-based things or three-path-based things for machine vision?
I think what I would like, I have to use the, yeah. I think more than anything, I think it's mostly on us rather than the AI people to develop data sets of behavior in more naturalistic settings so that we can train AI models that can do different behaviors, basically. And I think when you are doing stuff like this, that when you actually train models for different behaviors, sometimes it actually shows you that it works better if you have a two-pathway model. I don't know, maybe a three-pathway model. There's evidence of, in the visual system, having three pathways, one for the dorsal stream for action, one for ventral stream for object recognition, and the middle pathway for social interaction perception, basically. So I think, but the problem is that we have a lot of good data sets of object labeling, object categorization, because they're easy to collect, but it's the work of Madine and a lot of other people who are trying to move us out of the screen. I think we just need good data sets. We don't have enough good data sets of natural behavior, natural interaction with the environment to help us develop the AI models that can tell us sort of more about whether we need 2 pathways, 3 pathways, and how they interact with each other. Yes? You're welcome.
So we are a bit, we're not doing great time-wise. So let's take 10 minutes break.